Hello everyone. This video is to show you how to process the data that you collected in the Bio 105 lab exercise 2 quadrat sampling. So here we have some example data. Each group has gone into their quadrat and recorded the species of trees present and the number of each of those trees. Now, what we want to do is be able to take this data and turn it into something that we can both visualize and that gives us an idea of what's going on relative abundance wise and get a Simpson's diversity index out of it. So we already have in these other tabs for you set up the calculations necessary to do it. So really all you have to do is go in, highlight the tree's common name and the abundance. So once we have it highlighted, you're going to right click or control click, and we're going to go ahead and copy that data. You can also do the same thing by hitting Command C or Control C. We're going to come over to this other tab. We're going to go ahead and we're going to paste in our values. You can also do this with Control P or Command P. So now what it's doing is it is giving us our relative abundance. So if you remember, that is calculated, and we can actually see it here. That is the number of individuals of an individual species divided by the total number of individuals. All right, so we've got the total number of individuals. This is just the sum of all those, and that gives us our relative abundance. All right, we'll come back to that in a minute. This column is just doing that number of individuals times the number of individuals minus 1. Because if you remember, we've got our Simpson's index down here, and that is 1 minus the sum of all of those ni times ni minus 1s divided by the big N, which is the total number of samples, times the n minus 1. So that gives us our Simpson's diversity index down here. Then what we're going to go ahead and do is we can clear our data that we added in. And the way you want to do that is you want to right click or command click, and you go down here and you want to do clear contents. Okay, that leaves all your formatting in place. If you delete, it'll move columns around and mess things up. So we had that first index. We're going to come over to our next one. Go ahead and copy that. Paste that in. Okay. That gives us the index for our next plot. So you can either type that in, or if you copy this, because it's a formula, if you paste it the normal way, we get this sort of thing. What you want to do is you want to paste it as just the values, which is this next one over. So right click, paste as a value. Okay, now that'll stay there if we change the other numbers. So we'll go back and we will do it one more time for our third one. And you can actually just paste right over what's in there if you forgot to get rid of it like I did. Okay, so now we have our third and final one. We're going to copy that and then paste in there. All right. So now we've got our 3 Simpson's indexes. And what it indicates is that 2 of our plots are really very close together as far as their diversity, but another one is much higher. Right? Now, it might give you an idea that higher diversity means that their conditions are different in that particular plot. Now, let's go through how to turn this into a graph. What we're going to want to look at is the relative abundance versus species. So we're going to go ahead and highlight our data over here, and we're going to go up to Insert, and we're going to do another one of our bar graphs. Okay. Now, That put our data in there. We don't have our labeling or anything the way we want it. So what we want to do is we want to either go here and right click on the graph itself and go to select data, or we can come up here up above and go to select data. Now we want our horizontal category to have our axis labels. So we're going to hit edit there, and then we're going to go over and highlight our names, our column of names. So now you can see that we have our species on here. Now there's a few things we want to get rid of. All right, so if we select the graph, we can go to chart elements, that is either there or there. And the one thing we want to get rid of is our chart title. Okay, and we want to add axes titles. So if we click here, all right, that adds in our 2 axis titles. So if we go ahead and select over here, this one is going to be relative abundance, PI, and this one is going to be tree by common name. Let's just call it trees common name. All right, and so there is our graph. This allows you to get an idea. You can see at a glance what the relative abundance of each of your trees is. And we've got all of our data and all of our axes labeled, so you know exactly what all of those numbers are. Thank you once again for listening today. We'll see you in lab.